Welcome to another lecture by MedicoMedics. This week's med exam preparation, antibiotics. We begin our journey into the world of antibiotics, starting with some key high-yield foundations that every medical student and curious mind should know, beginning at the molecular level inside the bacterial cell. The ribosome, the cell's protein factory, offers our first clue into how antibiotics can selectively target microbes without harming us. Ribosomes make proteins by reading messenger RNA instructions, a process called translation. And these ribosomes are made of two parts: the 30S subunit, which reads the messenger RNA, and the 50S subunit, which links amino acids together to build the protein. Now, in bacteria, this machinery forms the 70S ribosome, indeed made of a 30S small and a 50S large subunit, while in human cells, it's an 80S ribosome composed of 40S and 60S parts. And it is this structural difference which allows antibiotics like tetracyclines, aminoglycosides, or macrolides to specifically target bacterial ribosomes without disrupting our own. And that's why these drugs are generally safe, unless in rare cases they interfere with mitochondrial ribosomes, which resemble those of bacteria. Now, the fundamental distinction between gram-positive and gram-negative bacteria lies in their cell wall structure. And this has massive clinical implications. See, gram-positive organisms have a thick peptidoglycan layer and no outer membrane, making them highly susceptible to antibiotics that disrupt cell wall synthesis, such as penicillins. Now, gram-negative bacteria, on the other hand, have a thinner peptidoglycan layer, but has an additional outer lipid membrane studded with porins, which restricts drug entry. And this makes many cell wall targeting antibiotics ineffective unless specifically designed to penetrate that outer layer. Now, antibiotics function by selectively disrupting vital bacterial processes, acting like precision-guided strikes. So some inhibit peptidoglycan synthesis, others damage DNA, some halt transcription, or interfere with some bacterial ribosome, shutting down protein synthesis. Now, each mechanism correlates with different drug classes, and knowing the specific cellular target helps predict both spectrum and side effects. However, bacteria can evolve. See, they pump drugs out with efflux pumps, they block entry by mutating porins, or modify drug targets like penicillin-binding proteins and ribosomes to avoid binding. These resistance strategies are why we use combination therapy in critical infections, for example. And an example of this is MRSA, or methicillin-resistant Staphylococcus aureus, where the A strain of Staphylococcus aureus has acquired resistance to methicillin and other beta-lactam antibiotics like penicillins. And without going into greater detail, we can just mention that the resistance here is due to the MAC-A gene, which encodes an altered penicillin-binding protein, PBP2A, that beta-lactams, found in our penicillins, can't bind with. So now that we've mapped out the bacterial battlefield and how the antibiotics strike, let's dive into the 10 essential antibiotic classes using our new mnemonic. Now, the enzyme is pills are surely the greatest cure for most bacterial infections. And we begin our journey at the bacterial wall. See, penicillins, members of the beta-lactam family, disrupt construction by targeting penicillin-binding proteins, specifically transpeptidases, the enzymes that link peptidoglycan like bricks in a wall. So inhibiting them weakens the structure, leading to rupture. Now, penicillin G was the original G, effective mainly against gram-positives. Later came amoxicillin, a smaller and broader spectrum antibiotic able to reach into some gram-negatives. However, not all walls fall. Like we mentioned before, we have MRSA, which has a modified enzyme, the penicillin binding protein 2A, which penicillins can't bind to. So it's penicillin resistant. Now, once past the wall, we enter the ribosome, the bacterial protein factory, and aminoglycosides bind the 30S subunit, not just pausing translation, but actually corrupting it, causing the ribosome to misread messenger RNA and produce dysfunctional proteins. And these aminoglycosides are especially effective against gram-negatives. But they need oxygen to be transported in, making them useless against anaerobes. Examples here include gentamicin and amikacin. These are the core drugs found here, and when paired with beta-lactams, which open up the wall, aminoglycosides gain better access. So it's a classic synergy here. Now, moving on, now we shut down the cell's ability to make DNA. And here is sulfonamides. Now, bacteria synthesize folic acid from scratch. That's unlike humans who get it from diet. So sulfonamides, like sulfamethoxazole, block the enzyme dihydrooctanoate synthase, and trimethoprim blocks dihydrofolate reductase. These are two critical enzymes in the pathway, or in this pathway. Now, without folate, no DNA, meaning no replication. That's why we combine them as TMP-SMX, a lethal 1-2 punch. And these are mainly used for urinary tract infections, for example, pneumocystis pneumonia, protozoal infections, for example. However, they must be avoided in people with glucose 6-phosphate dehydrogenase deficiency or late pregnancy and neonates. And that's due to risks like kernicterus. Now then, moving on to tetracyclines. So back at the ribosome, tetracyclines take a different approach. See, they also bind to the 30S subunit. But instead of corrupting the message, like the aminoglycosides would do, they block the transfer RNA docking site, so halting protein assembly before it starts. And the result is bacteriostatic action. You can compare that to aminoglycosides that have bactericidal action. And here we find doxycycline, tetracycline, for example. And these are widely used for treating acne, Lyme disease, chlamydia, rickettsia, for example. And they're broad spectrum, covering gram-positives, gram-negatives, and atypicals. However, they're avoided in children and pregnancy due to effects on teeth and bone. Also, don't take with milk or antacids. They bind the drug and block absorption. Just a tip. Now then, let's revisit the wall with glycopeptides. But this time, let's revisit it from the inside out. See, glycopeptides like vancomycin, they don't target enzymes. Instead, they bind directly to DLA tails on peptidoglycan precursors, preventing the wall from being built in the first place. And this shuts down construction entirely, leading to cell death. They're only active against gram-positives, though, since their large size keeps them out of gram-negatives. And vancomycin is essential in treating MRSA, or Clostridium difficile. So this is one way to go around the development of antibiotic resistance to penicillins. We have the same end result, essentially inhibiting cell wall synthesis. But instead of going at it from the outside, here we go from the inside. Now then, let's move on to cephalosporins. See, cephalosporins are beta-lactams too. They block the penicillin-binding proteins and disrupt cell wall cross-linking, just like penicillins do. But what makes them unique is their generation-based evolution. So we have first generation, third generation, fourth generation, etc. Whereby the first generation, for example, cefazolin, is best against gram-positives. Third generation, like ceftriaxone, is more against gram-negatives and also has central nervous system penetration. Then we have fourth generation, like cefepime, where you can add now treating Pseudomonas as well. Now, these cephalosporins are mainstays in treating UTIs, pneumonia, meningitis, and for surgical prophylaxis. Now, continuing on, we have our fluoroquinolones. See, fluoroquinolones hit the core. They hit DNA replication. Drugs like ciprofloxacin and levofloxacin inhibit DNA gyrase and topoisomerase 4, the enzymes needed to coil and uncoil DNA. Without them, the strands tangle and snap, like trying to unzip a knotted rope. Now they have a strong gram-negative and some gram-positive coverage, plus atypicals. For example, ciprofloxacin covers Pseudomonas, while levofloxacin adds respiratory pathogens as well. But be cautious, these drugs can prolong the QT interval, can cause tendon rupture, and are avoided in pregnancy and children due to cartilage toxicity. Next, we have the macrolides, like azithromycin and clarithromycin, and these bind the 50 subunit ribosome, blocking the exit tunnel so new proteins can't leave. So the factory keeps running, but the product jams, halting growth. Now, these are bacteriostatic and quite useful against gram-positives and atypicals like Mycoplasma, Chlamydia, Legionella. Now, azithromycin is favored for its long half-life and fewer drug interactions. But be aware of QT prolongation and note that clarithromycin and erythromycin inhibit CYP3A4, which can complicate other medications. Continuing to beta-lactams, broad. So here we find drugs like carbapenems and monobactams. And these target penicillin-binding proteins just like other beta-lactams, but they resist most beta-lactamases, making them powerful tools against resistant organisms. Now, carbapenems cover almost everything: gram-positive, negative, anaerobes. Aztreonam only targets gram-negative, but it is safe for penicillin allergic patients. Imipenem must be given with cilastatin to prevent breakdown by renal enzymes. Here, watch out for seizures, especially in patients with CNS disease or renal impairment. And finally, we have our imidazoles and rifamycins. So drugs like metronidazole and rifampin, they take out the genetic core. So metronidazole enters anaerobes, becomes activated, and generates DNA-damaging radicals. A very precision strike in oxygen-poor settings, like in abscesses, for example. Rifampin blocks RNA polymerase, halting transcription, and it's key for treating tuberculosis, for example, and meningococcal prophylaxis, and as a meningococcal prophylaxis. Now, use metronidazole for anaerobic infections and protozoa, like Giardia or Trichomonas. Rifampin turns, here's an interesting thing, rifampin turns body fluids orange and induces liver enzymes, which can reduce efficacy of other drugs. We've now journeyed through the bacterial cell, wall, ribosome, and genetic core. Guided by the mnemonic, pills are surely the greatest cure for most bacterial infections. See, each class targets a weak point, and understanding their mechanisms is key to smart prescribing and antibiotic stewardship. And that's the end of this lecture. Hope you enjoyed it. Please share, like, and subscribe for more weekly videos.